Thank you everyone for coming here. 안녕하세요. 감사합니다 for joining me on this sharing session. I'm going to go talk a little bit about our spatial portfolio, focusing in on the Cosmix as well as our ultra high-plex Cosmix panels. All right, so let's go through some of the material that I have for you today. So at Nanostring, when we were talking about the study of biology, we do believe that biology happens across multiple scale lengths and isn't really confined to, any one particular scale or any one particular cell type. We believe that biology happens across, for example, cell-cell interactions, across at the molecular level where you look at enzymatic reactions, but it also occurs at organ and tissue levels, right? So when you have a complex system like that, what you really need is multiple technology solutions that is able to come in at various length scales, various ways of profiling your biology to then give you a proper full depth insight into the biological systems that you're working with. So just a brief recap of the company. Company was actually founded in 2003 out of Seattle. It started off as a spin-off from the Institute of Biosystems Lab out of UWash. Okay, and then we launched our first instrumentation back in 2008, which was a bulk sequencing platform, the Encounter system, as well as 10 years later, the first spatial profiling system, the Geomx platform, that is really designed to go after cohesive units of biology. And slightly over a year ago, in the end of 2022, our Cosmix single-cell spatial molecular imager systems. The company today has 500 employees spaced out globally across the Americas, Europe, as well as APAC. Our annual revenue, is about 140 to 150 million USD per year. And currently we are about 7,000 publications and growing. So if you look at our publication track record, every day we get notification of 3 new publications coming into our portfolio. So one thing nice about all of our systems is that at the time of its commercial launch, the Encounter, the Geomix, and the Cosmix systems, they were all featured in Nature Biotech, right? And this really speaks to the disrupting and groundbreaking nature of the technology back at the time where it was launched. If you dive deeper into our spatial portfolio, right, just on the Cosmix chemistry alone, we invested 60 million USD. Okay, currently we have close to 150 publications going through a peer review process. Some of them have already been published, with many more in the pipeline. And one thing that we think about when coming up with technology solutions is that we always think about end-to-end workflows, starting from sample prep all the way to data analysis, right? And when it comes to sample types, we really believe that FFP compatibility is the key to having a successful technology that can then do clinical translational work. Because if you look at the clinic, the biggest repository of archival samples really is the FFPE sample type. So then how do I mentioned about multiple length scales, right? So how do we really think about multiple length scales? For example, if you take a look at a cross-section of a mouse brain, and if we zoom in at the hippocampus region of the mouse brain, so the hippocampus is broken down into many discrete sub-regions in the brain. So highlighted here, okay, let me change my pointer to make it a bit more obvious. Highlighted here, you have different regions of the hippocampus structure. And at the bottom, this little wishbone-like structure is the dentate gyrus, right? So the dentate gyrus is important because it is the structure that's important for forming new memories. So one thing that you can think about is, if I were to just profile areas like dentate gyrus, specific regions of the hippocampus like the C3, the C1, and basically I could do this for a whole patient cohorts. What I can then easily do is to pan between these different patient cohorts and see whether differences in ability to make memories, differences in ability to process memories, store and retrieve them, right? Or even neurological pathologies, whether or not differences in biology between these sample types have any implication of neuropathologies. So on a broad level, this gives us a very robust tool to very quickly pen through cohesive units of biology and compare them across different study arms or different cohorts that you have across your patient types. So that's what the Geomx is able to do. And with the Cosmix, we're able to dive even deeper in resolution, in the spatial resolution of the system, and start looking at single cell, single molecule level details of what comprises each cohesive unit of biology. So while the Geomix platform tells you what is happening, the Cosmix platform would then go into each discrete phenomena and tell you exactly why it's happening and who is participating in it. So the Cosmix, you then know the drivers, the individual cells that make up each different class of disease, whether it's a person who's able to make new memories or a person who isn't, what's the differences in cell types that are being recruited into the dentate gyrus, what are the differences in the signaling pathways that are being upregulated in the dentate gyrus, right? So in a way, to summarize, the Geomix will then tell you what's happening, the Cosmix will then tell you who did it and how. At a very broad level, how we differentiate between these two platforms, aside from the scales of the data that's being processed. So with the Geomix, you have very high sample throughput. Data is output in a very simple count table format, which makes it easy for you to process and analyze for clinical applications. For the Cosmix, because we're really looking at a 50 nanometer resolution scale of all samples that we process, we're really limited in terms of the types of throughputs that we can get out of the instrument. And this is limited to 4 to 8 sections, depending on the plexity of your experiments and the amount of tissues that you're analyzing. But in exchange for slightly lower throughput, what you get is unparalleled resolution, 50 nanometers in the X, Y, and Z axis, right? And all of this without sacrificing anything in terms of the plex of the experiment. Just a quick recap of Geomix before I dive deeper into Cosmix. So with the Geomix, we have very high plex panels, okay? And these start with our protein assays where we have anchoring our IO proteome atlas, which provides in a single assay the ability to assay more than 570 different protein targets in a single Geomix experiment. Right. In addition to that, we also have smaller protein panels that allow you to customize your approach to analyzing your science. On the RNA portion, we have whole transcriptome panels for both human and mouse, as well as more targeted panels like the Cancer Transcriptome Atlas, the Canine CTA Atlas, as well as the RNA immune pathways. And if none of these really suit your research needs, we also have the ability to provide full customization of both of our protein as well as RNA assays. So what exactly is the IPA assay or IO Proteome Atlas? Essentially what we did was partner up with Abcam and translated their whole entire IO proteome catalog into a single assay, right? So what this does is that we took 570 different protein targets, okay, and we validated it across multiple different internal and external validation trials. And these 570 different protein targets covers thematically cancer, metabolism, infectious disease, organ transplant, epigenetics, autoimmunity, anything that can be implicated in oncology, it will target it. Because of this, it also provides comprehensive coverage of the hallmarks of cancer paper, as well as 77 different functional pathway annotations within the panel. Right, and to this panel, you have the ability to add on an additional 40 protein targets of interest. So the entire panel has been validated extensively with our partner Abcam, where we do single plex type validation for pre-post conjugation to ensure that conjugation for our Geomx labeling reagents doesn't change the specificity of the antibodies that we're using. In addition, we also do multiplex validation internally by Nanostring using FDA-approved CPA and TMA assays. Right? And from here, this will then allow us to understand whether or not there's any crosstalk or cross-hybridization between targets. So all of these data sets are available publicly on our website. You're more than welcome to download it to view the data from our IPA assay yourself. So one nice thing about IPA assay is that it can be combined with our WTA assay to then provide in a single experiment, single sample, single assay, both whole transcriptome level of RNA coverage, as well as ultra-high 570-plex protein antibody coverage. Right? And then this would allow you to bring your science closer to clinical discovery because it takes out sometimes the guesswork when translating RNA into proteins, because usually there isn't a one-to-one translation, taking into account things like post-translation modification, activation, and degradation. So if any of you would like to see further what Geomx data, Geomx images look like, feel free to explore the spatial organ atlas hosted at the Nanostring website. All right, so this covers multiple different tissue types for both mouse and human. All right, not going to go too far more into Geomx because everyone's here about Cosmix, but like I mentioned, we have papers coming in every day, and this is for both Geomx and Cosmix. And even for the Geomx platform, we still get very high impact papers coming in every day. Things like Cancer Cell, not too long ago. All right, so that's my short little spiel on the Geomx platform, and let's then go on and start talking about what Cosmix really looks like, and then what does genomic graph do for you on Cosmix platform. So just a quick overview of what your typical Cosmix experiment looks like. Every single Cosmix experiment, you would expect to generate about 500,000 to a couple of million cells, right? And this is at 1,000 plex to 6,000 plex, currently possible transcripts, gene targets that we assay across every single cell. What this really results in is that for any single Cosmix experiment, we are looking at billions of data points. And if you take into account what this number really means, like taking number 4 million, this is roughly the number of banked cells, single cell data in the Human Cell Atlas project from its inception all the way to 3rd of June 2022. And if we follow how much single cell data the Cosmix has generated right now, we're roughly closing in on 4 times the amount of banked single cell data in the HCA project. Right? And this is for a single Cosmix run, 15% from a single Cosmix run. So what exactly is Cosmix? Right? Cosmix is basically a way for us to go in and detect with single molecule level of accuracy individual RNA transcripts present in your tissue. To help drive data analysis and cell segmentation, we also have the best-in-class cell segmentation models that are driven not only by DNA staining, but we actually also go in and stain for cell membranes. We stain for the cytoplasm. So it's the combination of the DNA, the nuclei of the cell membrane, as well as the cytoplasmic staining that then allows us to train the best-in-class cell segmentation module out there amongst all of the commercial single-cell imaging platforms to date. We're also the only true multi-omic platform having a capability to provide right now, what we have shown is whole transcriptome level of RNA coverage together with more than 120 plex protein antibodies. All right? And like with all of our other platforms, when we first designed the technology, we designed it from grounds up to be compatible with FFPE samples. So what this means is that all of our workflows, they're intended to be similar and compatible with your standard IHC workflow in the lab. We don't do anything fantastic. We don't do anything different. We don't even use special microscopy glass for our experiments. We just use the standard workflows, standard microscopy glass, standard FFPE blocks. So for all of you single-cell researchers out there, this kind of t-SNE plots, UMaps would be very familiar to you. And in a way, it marks sort of the endpoint of your single-cell experiment. However, no amount of algorithmic manipulation would then allow you to recreate the exact tissue type that these different disparate cell types came from. What this means is that a lot of the inferences that you make out of a single-cell dataset are purely inferences. We go at it based on what we understand, our understanding of canonical biology, based on our best understanding of how cells like to relate and cluster with each other. And really, the whole purpose of the Cosmix platform then is to remove the guesswork, remove the inferences out of doing any form of single-cell in situ study, and basically allow you to study your cells in the tissue where they are taken from, without having to dissociate any tissue, without having to perturbate your sample, without having to change your system. So this is some of the assays that we have built around the Cosmix platform, all right? Some of which are currently available. Things like the human immune oncology panel, the mouse neuroscience panel. Coming soon would be the human neuroscience panel, as well as the mouse immune oncology protein panel. In terms of RNA assay, we have both 1K human as well as mouse universal cell characterization panel. We also have an additional mouse neuroscience 1K panel that's already available, as well as a human 6K discovery whole reactome panel. Coming soon would be the mouse version of the 6K discovery panel, as well as the human whole transcriptome panel. And we expect these to be available for the whole transcriptome to be available in the later half of next year. For the mouse discovery, we expect that to be available sometime towards the end of this year to early next year. Same with our Geomx panels. There is always the ability to do a custom, whether it's in the form of a spike-in to an existing panel, or whether it's a full standalone panel that you would use. So let's take a step back. Let's think a bit deeper about what are some of the challenges that single-cell researchers face in clinic. So if you think about what a single-cell workflow starts and looks like, it starts with first taking a significant chunk of tissue or sample and dissociating that. And usually your sample that you take would have anywhere starting from 200,000 to millions of cells to begin with, out of which you're only really sampling 5,000 to 20,000 cells. So when you're sampling at roughly 5 to 10% of your input population, how confident are you that you're able to catch that rare cell type that presents itself at 0.1% of your population? Right? Again, because you're not sampling a lot of data points in your samples, you only really get partial cell coverage as well as partial coverage of the cell states of the cells that are within your sample. Furthermore, this is complicated once you start moving into FFPE type samples because the RNA is degraded and therefore is not very amenable to any form of library preparation. Another thing that you would be familiar with is this thing called the doubling rate, where doubling doublets in your single-cell experiments can account for up to 30% to 40% of your data if you're not careful with how the single-cell experiment is done. This is coupled with the high cost of sequencing, complexity of data analysis, as well as the difficulty of scaling, right? Having said that, I do acknowledge that there are new emerging technologies coming out in the market that allow you to sequence 2 to 3 million cells. But at the end of the day, you are still plagued with things like the having to dissociate your sample, complexity of data analysis, as well as high sequencing costs. Let's take a look at what Cosmix can offer your traditional single cell researcher. First and foremost, we profile every single cell that is present on your specimen, right? There is no subsampling. And this then gives you confidence that whatever area we study, if the cell is there, we will see it. Whether it's an ultra rare cell or whether it's something that is very abundant. You don't have to worry that your data is being skewed towards the abundant population and you're going to miss out that rare cell populations. We are built from grounds up for FFP samples, and you'll see in a bit what that really means once you start looking into sample quality. Furthermore, the instrument is an end-to-end workflow. There's no need for the further downstream sequencing required. So that gives you immediate cost savings from not having to sequence your samples. All of our data is handled informatically using our Atomics cloud platform that provides easy to use one-touch analysis solutions that even a non-informatician can use to drive simple, high-level insights from your samples and your experiments. So all of these combined allow us to have unparalleled scalability. So what exactly is the 6K platform? 6K platform covers more than 6,000 different RNA targets, right? And why we call it a discovery panel is really because it provides a whole reactome coverage for the human reactome. Together with 6K panel, we have best-in-class multimodal cell segmentation. So for cell segmentation, there is the ability to use a mix of nuclei staining, membrane staining, cytoplasmic staining, right? And if we can't find the appropriate antibody, we also have the ability to do transcript-based staining to allow us to help with complicated cell segmentation, right? And this is compatible with a wide range of tissues that we have already tested in-house, up to 57 different tissues currently. This is currently available only for human samples, but the similar mouse panel will be coming soon at the tail end of this year to early next year. And there's always the ability to add on an additional 200 custom gene targets. If there are specific molecules that are of interest to you, they are not within our 6K panel. So we mentioned a lot about genomic graph. We mentioned a lot about whole reactome. This is what a whole reactome high genomic graph panel looks like. So this is what we call a Voronoi diagram. You don't have to know too much. It's basically a cute way of drawing polygons. But what's important with this map is that we're just essentially, we have listed out thematically every single pathway or every single pathway that is represented in the human reactome. And what we did was coloring these pathways based on pathway coverage. You can see that essentially we have whole reactome coverage with our 6K panel. Now imagine you're trying to do this type of discovery work with a low plex panel, something like a 380 plex panel. What you're going to see is that coverage is going to look a lot poorer. You're going to get mostly white space within the reactome map. And then the question becomes, what are you missing when you're doing a low plex panel? So just some examples of how we have already deployed our 6K panel, all right? So some of the data that we're showing is a mix of in-house data. Some of it will be from customer data that is either going through peer review or is going to be pre-publication, still at R&D phase. So this is a poster that we displayed at SITC, and this really shows. This poster is really talking about squamous cell carcinoma and some of the interesting findings that we have. I'll talk a little bit more about this, but if you want this poster, it is hosted on our website. You can go read more into it. So we do have hosted on our website as well a human brain data set. Again, this is something that you can freely download. It's open to everyone. We don't ask where you come from or what you do with the data. We just ask that you do something meaningful with it, right? So this is a data set that you can freely download from our website and use it to manipulate to your heart's content. and from there start exploring at the types of data that you can get out of a high-plex platform like the Cosmix. So one of the cool things that we saw out of this 6K human brain dataset is that when we start looking at how receptor-ligand interactions cluster, what we noticed was a very interesting subcluster of receptor ligands, basically the SST, CORT, as well as NPY, and we do see these molecules cross-talking with each other. What is also interesting is that when we start co-localizing all of these molecules, they're essentially co-localizing to the same cell, the SST trundle cell, right? And these cells account for less than 1% of the brain in terms of number of cells. So what we've done essentially with this experiment is not only have we pulled out a very rare cell type, we have also showed very interesting biology happening in this cell type, the SST trundle, which happens to also be one of the biggest neurons that you have in the body, covering vast distances. All right, let's go back a little bit to the skin project. So what's nice about the 6K panel is that it essentially allows us to subdivide cancer or our squamous cell carcinomas into 8 specific distinct subpopulations of cancer cells, right? Not only that, we can also then correlate this, the cells that we have identified together with marker genes, right? To allow you to validate your work as well as see the spatial distribution of these cells and marker genes that then drive the different cell lineages. But what's really interesting about this work is that what we're seeing is radiating out from the keratinous pole of the squamous cell carcinoma, we see cancer cells in different states, different lineages. And all of this is only possible because we're able to come at this type of tissue with an ultra-high-plex panel. And this then allows us to go after things like lineage tracking in cancer. It allows us to better understand perturbations in therapy. And then it also allows us to understand resistance in the clinic. So one of the really cool things that we have seen for this particular set of samples really is in terms of how macrophages respond to keratinocytes in the tumor. And specifically what we have identified is a very distinct phenomena of monocytic recruitment to where you have your tumor nest, as well as a driver towards macrophage differentiation that's happening across the samples, right? Right. So let's talk a little bit about comparisons between different platforms, right? I think the field has been developed enough over the last 12 months that when you start talking about single molecule, single cell imaging platforms, you do have an overabundance of choice, right? But are all single molecule imaging platforms really equal? Let's take a look at some of the comparisons data that have come out from our customer base, right? In this case, first I'm going to talk about Dr. Prambir Dulai out of Northwestern. So Prambir here, he did a very comprehensive study looking at 16 different blocks representing 11 different patients for ulcerative colitis as well as Crohn's disease. What he did was he took these blocks, cut out serial segments out of the blocks, and immediately after sectioning, processed them simultaneously on the Cosmix SMI in his lab, as well as the Xenium in the spatial core that is located in Northwestern. So these experiments were running simultaneously head to head. And after that, he took the data out of each platform and did a comparison of the data that came out of both platforms, right, on adjacent samples performed at the same time. So some of the key findings that he found out was firstly, Cosmix detects 4 times more transcripts and 6 times more genes per cell with more consistent cell typing. And that really what drives all of this is the importance of having high quality cell segmentation. And the fact that our chemistry is built from grounds up to be FFP compatible really shows in the consistency of performance of Cosmix platform, regardless of block quality. So by the way, this seminar was hosted on Genome Web. If you want to view the seminar, please feel free to scan the QR code after this. I will be providing the deck to the Filicoria team, so they will disseminate the deck to you for your reference. So just some high-level metrics that we have seen come out of the Cosmix versus the Xenium. So in terms of plex, Premier used the 1000 plex panel without the add-on. So how our 1000 plex panel is designed is that it has a 950 core module together with a 50 plex add-on. Together, they will give you your 1000 plex panel. Premier only used the core module, that's fine. And he compared it against the Xenium the Xenium pan cancer pan tissue panel that stands at 377 plex. So these are just some high-level statistics that he got out of the different studies. Out of the Cosmix, he got about 185 transcripts per cell. Xenium roughly at 46. And if you look at the ratios, that roughly is a 4 times ratio of Cosmix over the Xenium. And this is something that cannot just be accounted for by the increase in genomic breadth, right? Our panel has roughly 2.5 times the genomic breadth of the Xenium panel, but use 4 times more transcripts per cell over Xenium. If you look at the unique mean genes counted per cell, we're also roughly 6 times more sensitive than what you get out of the Xenium. But at the end of the day, in a way, we are biasing the study because we have a much higher plex panel, right? So let's say, what if we leveled the playing field a little? What if we only look at common genes, right? Let's discard our advantage of genomic breadth. Let's just look at common genes. So we did a correlation plot, plotting sensitivity for Xenium against Cosmix. What you see is that the majority of samples showed higher sensitivity on the Cosmix platform over the Xenium platform. And this is us discarding our advantage of genomic breadth, going down to the Xenium's plex and saying, let's be comparable to you. What does that really mean? Even with a reduced plex, the Cosmix outperforms the Xenium for overlapping gene content. And if you start looking at cell size and cell segmentation, on the Cosmix platform, you get cell size ranges that are more in keeping with normal biology, as opposed to, for the Xenium, when you start looking into stromal cells, immune cells, you start seeing inflated cell sizes creep in, like your T cells having a 300 square micron cross-sectional area, your glial cells, your stromal cells, your fibroblasts. And this is important because when you start thinking about how cells are segmented, if they're not segmented accurately, what you're getting in your data set is essentially a lot of noise from surrounding contaminating cells. And you can think of this in your single cell context as your doublet rate or even your triplet rate. And normally in single cell, we're able to accurately spot out doublets or triplets, and we know to prune them from the study. Unfortunately, for single cell in situ spatial type data, the picture isn't that clear. You would have to informatically manipulate your data, find out the contaminating signal, and then prune it out yourself. So we talk about consistency of performance of Cosmix regardless of sample quality. So one thing that Premier did was, in addition to performing the experiments head-to-head simultaneously, he actually also profiled the RNA integrity or RNA quality of each of these blocks that he sent for Cosmix as well as Xenium profiling. And what he found was that the blocks had a range of DV200 scores. Some of them had decent DV200 values around DV240, but a lot of them were below DV200 40%. What we see is that the Cosmix had more or less consistent performance regardless of RNA quality, as opposed to the Xenium, where you start seeing a drop-off in your counts as the quality of the block degrades. And if you were to follow best practices for Xenium, Xenium does recommend only running samples with a DV200 score of more than 30%. All right. So that is the difference between a technology that has been built grounds up to be FFP compatible versus one that was built for fresh frozen and then adapted for FFPE samples. So just to drive home the point of cell segmentation, this is not an individual observation from Prambir, but this has also been corroborated by Dr. Suzuki from UTokyo from an earlier study that was published in December last year. So what Dr. Suzuki did was he looked at breast cancer samples on both the Cosmix as well as the Xenium, and he saw the same phenomena where on the Xenium, you're getting wildly inflated cell sizes, especially for cell lineages that co-locate to the stroma of the samples. So even accounting for wildly inflated cell size, if you look at gene by gene sensitivity in matched FOVs, it is similar across both Cosmix and Xenium. But the question is it truly similar? If you remember earlier in the year, in 2023, we had this whole conversation about what FDR really means for spatial biology. But also, what's more important in spatial biology is really the phenomena of having accurate best-in-class cell segmentation. So this is a study that was published out of the Satija lab. And what they did was they analyzed data from different spatial platforms. One interesting conclusion that they came up with was that on a certain platform X, cell transcript counts were inflated up to 3 times on average. So then what does FDR really mean when noise accounts for up to 70% of your single cell transcript data for every single cell on average? What does that really mean? Right, so that's something to think about. And if you take that into account with some of the earlier charts that I showed, things like the sensitivity, things like cell counts with decreasing RNA block quality, how much of your counts are you really going to lose once you start pruning into data that is just noise versus data that is true biology, right? So, like I said, Cosmix is the tested and proven platform. We have quantified more than 57 different tissues on the platform today. And we also have partnered with some of the leading academic and commercial institutions globally. And all of this is anchored by our Atomic Spatial Informatics platform that allows even non-informaticians to derive high-level insights from their samples. Of course, at the end of the day, you would still need to consult an informatician in order to make the most out of your data. But if you need some quick and fast insights into your samples, Geomx is a robust platform that allows you to do both data manipulation as well as data visualization. So lastly, to iterate, at Nanostring, we really believe that biology happens at multiple scales, and therefore, in order to get to the root of your biological question, you really need the capability to perform biology at any scale. Right, and that is the end of what I have to say.
Okay. Thank you, Jen. Thank you for your wonderful presentation about spatial biology and especially the Cosmx. And